Okay, this video is about immunoexcitotoxicity and microglia, mechanism of brain damage. And this is a mostly new talk since my recent talk about phosphatidylserine externalization with regard to causing brain damage. Okay, we're going to talk a lot about microglia. Microglia are the macrophages of the brain. That means they're the security guards of the brain. In a resting phase, they look like an octopus. They got these big tentacles sticking out of them, kind of amoeboid looking. Okay, as they start becoming activated, initially the first stage is called priming. Then they kind of look halfway between a fully activated over here and the initial resting one, meaning that they have much less ramifications of their tentacles, so to speak. Again, they look like an octopus in their resting phase. As they're exposed to more toxins or higher doses, more intense forms of stress or problems like infections, traumatic brain injury, glutamate, inflammatory chemicals, IL-1, TNF-alpha, autoantibodies from autoimmune disease, they become more fully activated. Once they're fully activated, now they start. So the primed microglia doesn't secrete stuff, but the fully activated microglia does secrete things. It secretes cytokines, reactive oxygen species, and it activates inducible nitric oxide synthase, and that ends up being a big deal. It's a different nitric oxide synthase than the one you see in the arteries, the endothelial nitric oxide synthase, the one that Caldwell Esselstyn talks about all the time, and Nathan Bryant. Okay, so anyways, this is going to be a key thing to understand about brain damage. And the reason you want to understand about brain damage is the more you know about what causes it, the better you are at avoiding it. A couple of little tidbits that we'll have to get out of the way. Cerebellum is the worst affected spot in autism. Autism, if you look at the brain neurons, the cerebellum is where about 70% of your brain neurons are located, even though it's a small little thing in the back. The reason is that's for fine motor coordination, tuning things. If you look at the cerebral cortex, the outer part, cortex means bark, like the bark of a tree, 90% of the cortical neurons have glutamate as a neurotransmitter. So glutamate is by far the most important neurotransmitter. It's an excitatory neurotransmitter. In other parts of the brain, it's more like about 50% of the neurons are glutamatergic. And so that's why you'll sometimes hear different numbers. Okay, there is a PET scan that has a special tracer that can be used to look for microglial activation. So when people hear a PET scan, what they're usually thinking is FDG, glucose, and that's used to look for cancer. But there are special research types of PET scans that can be used to look for microglial activation, meaning that they're sort of inflamed. Okay, here's an example of what a synapse looks like. And we're going through this just so everybody's real familiar with it. I'll be brief about it, but here's a presynaptic neuron, postsynaptic neuron. Here is the astrocyte, the supporting cell. So these three cells, pre, post, and astrocyte, this is your typical tripartite synapse. There's a fourth cell over here that's going to be relevant for us. It's a microglia. The microglia, again, looks like an octopus. These are the security guards of the brain. And we'll talk later about how they can get involved, but you should have this concept in your mind's eye. 1, 2, 3, 4. That will be useful. So increasing calcium in the cytoplasm from opening up of voltage-gated calcium channels, that will cause a glutamate to diffuse across the cleft and activate the postsynaptic neuron. Cortisol from psychological stress, sleep deprivation, caffeine, excessive MSG, MFG, manufactured free glutamate, even excessive sodium can activate the sympathetic nervous system, fight or flight system, and that will increase cortisol and will increase then this release of glutamate across the synaptic cleft. Okay, these are the NMDA receptors. They got magnesium in the center. They are a channel for calcium. Here's the AMPA receptors. They're a channel for sodium. Okay, and these are the synaptic receptors in the synapse. The ones out here on the side are extrasynaptic. So this would be the extrasynaptic NMDA receptor for glutamate. Okay. And again, the way it works is the dendrites are the incoming information to a neuron. When it's activated, it'll release an action potential beginning at the axon hillock, conducted by sodium channels till it gets down to the axon terminal. Then this depolarization, meaning increased positivity of the resting membrane potential, now it's the action potential, membrane potential, will activate and open up the voltage-gated calcium channel that lets calcium in. The calcium increase in the cytoplasm causes the neurotransmitter to go to the synaptic cleft, diffuse across, activate the postsynaptic neuron. So that's all basic neuronal transmission. Here's just a little bit of what a brain cell looks like. Calcium is always much higher, about 15,000 times higher outside the cell, extracellular matrix, than it is inside the cell. So in order to pump against this gradient, you need ATP, meaning you need to use energy to pump against the gradient. Okay, and that can be coupled to what's called the NACA exchanger, NACA, sodium calcium exchanger. In addition, you can pump calcium into the endoplasmic reticulum. It's called the circa because the S sort of hangs on for sarcoplasm, which is meaningful in muscle but not important in neurons. And these are the circa pumps. They're also ATP dependent. Then there's a slow channel pump over here, plasma membrane calcium ATPase, but that one's not that important. This one is much, 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 much faster. Okay. And here's just a sample example of excitotoxicity. If you release too much glutamate going across the synapse, it'll activate the AMPA receptors. They're really primarily a sodium channel that will depolarize, meaning make it more positive, the membrane potential, not resting, just membrane potential. Once this becomes more positive, the magnesium block, the magnesium in the center, it's an ion in the center of the NMDA channel will bounce out of there because it no longer has a negative charge inside here to activate it. At rest, you're about negative 65 millivolts. Anyways, other things that can open up the NMDA channel besides just binding glutamate, it can bind a few other things, including serine, and I'll talk about that a little later, but there's other things that can activate it. This is stuff we already talked about, and how the extrasynaptic NMDA receptors are the ones especially prone to, when they're overactivated, inducing excitotoxicity. This one tends to usually be a positive thing and is mostly associated with memory. When you have elevated cortisol, like from psychological stress, sleep deprivation, or drinking caffeine, you're going to get an increase in blood lipids, increase in blood glucose, so it causes insulin resistance, but especially important for our purposes, you're going to get increased glutamate in the hippocampus. Okay. I know some of this you've seen before, but trust me, I'm moving into more advanced stuff, but I want you to have the basics there before we get into the more advanced stuff so it'll all hold together. When a microglia is activated, it can release glutamate, and that can cause excitotoxicity of the neurons here. It can also release, reactive oxygen species. It can also turn on inducible nitric oxide synthase. I'll show that all in a moment, but those are some of the things it can do. I'll have better pictures. This is just a book review lecture I gave recently showing that MSG, MFG, they will increase plasma cortisol. So there's sort of a vicious cycle going on where cortisol increases glutamate transmission in a synapse. But what this is saying is glutamate, like dietary glutamate in the form of MSG, MFG, that will increase cortisol. So that's a potential vicious cycle, and it's another reason why you don't want to eat that stuff. Okay, as far as a typical plasma membrane in a neuron, at baseline, you want to keep the PS, phosphatidylserine, on the inner leaflet of the plasma membrane. The cytoplasm leaflet is the same thing as the inner leaflet. And the cell does that using ATP. The enzyme is called flipase. Simultaneously, you're going to push a phosphatidylcholine to the outer leaflet, and so that's called floppase, the enzyme that does this. Both of them are dependent on ATP. So the neuron has to be making adequate ATP in order for it to run these flipase and floppase, and that's normal. The key thing is phosphatidylserine is along the inner leaflet, cytoplasm leaflet. If you then have some damage to the cell that's going to make the cell become non-viable and go into apoptosis, for example, programmed cell death, this enzyme scramblase can flip them in the opposite direction and put phosphatidylserines on the outside. Then complement proteins will bind to the phosphatidylserines, and that's a signal, like a big sign that says, eat me, and then the macrophage, the microglia, will come in and it will eat the neuron. And in small amounts, this sometimes can be useful for synaptic pruning and things like that. It's important during development, but it's not good when a normal neuron gets eaten for the wrong reasons. Okay, there's a bunch of things that can lower the ability of the cell to make ATP, and that especially is going to be things like, lack of oxygen, a lack of glucose, hypoperfusion for whatever the reason, intracranial atherosclerosis, causing a chronic decrease in the oxygen and glucose ability to be delivered to the brain cells, the neurons. Mitochondria inhibitors, so they're going to decrease the ability to make ATP. The problem is if you start dropping your ability to make ATP, then you can't run flipase and you can't keep your phosphatidylserines in this inner leaflet, and they start going to the outer leaflet, and then they become an eat me sign for the macrophages and the cell dies. So that's why neurons have to maintain their energy, which is why you want to eat in a healthy way, so it's more easy for your body to do that, okay? Usually your liver, after you eat dinner, you can maintain your blood glucose for about 2 days just based on the liver storage of glycogen and gluconeogenesis. After that, beyond 2 days, they start going into ketosis, and you'll have ketone bodies made and whatnot. Other things that can cause problems if you can't make ATP because your mitochondria are inhibited. If you can't pump out calcium from the cytoplasm as fast as you want because the circa pump's inhibited. Excessive stimulants mean that more and more glutamate keeps pouring in, so everything has to be even more intensely working in order to keep that calcium in the cytoplasm from not getting too high. Okay, we talked also about you can have, you eat too much dietary fat, you get insulin resistance, you use up your insulin degrading enzymes, so there's none left to clear out beta amyloid. It not only clears out insulin from the blood, it clears out beta amyloid. Beta amyloid goes from the periphery, the outside part of the body, not the brain and the spinal cord, into the brain, and it'll bind to a microglia and activate it. Now the microglia will almost think it's a bacterial infection and will release toxic chemicals like reactive oxygen species, ROS, to sterilize the area. But that so-called sterilization process will damage the neuron. And then damage to the neuron will cause phosphatidylserine externalization, and the microglia will eat it. That's sort of a quick summary of what happens. Okay, if you got leaky gut, you're going to get bacteria, especially bacterial endotoxins across the leaky gut. Those can get into the brain because there's usually a leaky blood-brain barrier when there's a leaky gut-blood barrier. And these can bind a microglia, the macrophages, and they can cause it to activate iNOS, inducible nitric oxide synthase, okay? So inducible nitric oxide synthase is different than endothelial nitric oxide synthase, the one in blood vessels, like in these blood vessels, because it stays on prolonged amount of time, like for hours, and it can release tons and tons of nitric oxide. Nitric oxide can do a lot of damage. I'll show you a picture in a little bit of what it can do, but it can diffuse into it's like a gas, it can diffuse into the mitochondria and then compete with oxygen for binding to cytochrome C and basically shut down the mitochondria. That's a disaster. Okay, we'll get to all that in just a moment here. Okay, here is just one paper about mitochondrial allostatic load, meaning the more things you have going on that are damaging your mitochondria, excessive saturated fat intake, okay, all these mitochondria inhibitor pills and poisons, lead, HG, AL, and a whole bunch of other ones, hydroxynonenal from eating omega-6 fats. They all work together, and this one author thinks that that's one of the major contributing causes to autism. I've heard other things that contribute to autism, including MSG and MFG. And I've heard other ones too, but they're thought to maybe be multifactorial. Okay, this is an important slide. Immunotoxicity of fluoride and of AL, and just the theory of immunotoxicity. And here's all the things that can activate the microglia, can prime them and potentially activate them. Stress, obesity, diabetes. So it's really bad to be fat and diabetic. Okay, stress, beta amyloid, ischemia, toxicity, meaning lack of oxygen, traumatic brain injury, infection, like with bacterial endotoxins, LPS, LTA coming through the leaky gut, autoimmune diseases, like where there's autoantibodies, pesticides and herbicides. The immune system is relatively immune privileged, so there's usually not much of those sorts of things around, and when they're present, they can activate the microglia. Herbicide in particular would be like something like glyphosate, glycine phosphate. These inflammatory cytokines, interleukin 1b, tumor necrosis factor alpha, heavy metals, lead, Hg, Al, and then you have F minus, from tap water, and you can also get complexes between F minus and Al that can be relatively surprisingly toxic. Okay, then I kind of mentioned those already. We talked about the synapses. Okay, that's good enough for that. Okay, here's just coming back to that one slide I showed you earlier of a resting microglia, like an octopus, then a prime sort of halfway in between, and then a full-blown activated microglia just being round. Okay, chronic neurodegeneration. If you get release of this inflammatory cytokine thing, tumor necrosis factor alpha, it actually damages the amino acid transporters, excitatory amino acid transporters, so it'll decrease your ability to clear glutamate from the synapse. That's bad. It actually leads you to making more glutamate. It causes problems. You don't want that. Okay, so when there's brain inflammation, things go wrong. Acute microglial activation, that will do a few things. It'll release more glutamate, so that can cause excitotoxicity. It also can release something called quinolinic acid, and that contributes to excitotoxicity. It can release the amino acid aspartate, which I kind of call baby glutamate. That can increase excitotoxicity. Aspartate also reminds me of aspartame. It's in aspartame, and that can contribute to this process. The big problem is the microglia will turn on inducible nitric oxide synthase, and then they're going to send all kinds of excessive nitric oxide over to the neuron, and it can do a lot of damage. And so anyways, this is interesting stuff. There are such a thing as AMPA receptors. So AMPA receptors are primarily sodium channels, okay? But when there's inflammation, the inflammation can convert the AMPA channels into something very much like a NMDA channel. So what I'm saying is normally it's only a sodium channel, and you've got something called glutamate A2 receptors, and they block calcium. But when you lack glutamate A2, you can switch out the type of AMPA receptors on the plasma membrane, and the ones lacking glutamate A2 will allow calcium entry, and they can increase excitotoxicity. Now, normally, why would a cell ever want to do that? Well, it might want to do that under normal circumstances when it's trying to memorize something, increase the strength of the signal. But it doesn't want to do that for no reason, because if you overdo it, you'll lead to excitotoxicity. So here are metabotropic glutamate receptors, and these are ones, so these are ionotropic glutamate receptors, things like AMPA and MDAR and kainate, versus metabotropic glutamate receptors. They activate a cascade of reactions. So just enough to know about them. We're not going to get into them too much. In general, they tend to be excitatory. There's more to it than that, especially, the group 1. Okay, a couple things that are a little interesting. The amino acid transporters, excitatory amino acid transporters, they're blocked by hydroxynonenal, which comes from eating omega-6 fats. So you really don't want to eat omega-6 fats. Lots of bad things happen from omega-6 fats. I recommend avoid them as much as you can. If you just eat plant food, you'll get some. You'll get the amount you need. You won't get more than that. If you eat processed food, you end up getting more than you want. You also get arachidonic acid, sort of a long chain omega-6, which is relatively inflammatory. You'll get that like from chicken and from eggs, and you don't get that from plants, which is another good thing about plants. You can also, like I said, when a microglia is activated, it can release reactive oxygen species, reactive nitrogen species. You can activate this whole inducible nitric oxide synthase, start inhibiting mitochondria. You go down this big path of inflammation that you don't want to go down. There's something called the cysteine glutamate antiporter, which, it's not that important. We're not going to get into it, but you'll heard of it. If you want to look it up, you'll find it here. It's a minor player in the big picture of things. Okay, so Al and F minus, they form complexes, and the F minus helps you to absorb more Al in the gut. It helps more Al to get into the brain. And then this is both in typical tap water. So these two toxic chemicals, Al and F minus, accumulate in the human body. And the more they accumulate, the more likely they can have a synergistic effect to cause some brain damage, okay? F minus inhibits a whole bunch of enzymes. It inhibits enzymes in glycolysis like phosphofructokinase and enolase. It inhibits glycogen synthase. It inhibits enzymes in Krebs cycle, succinate dehydrogenase. It inhibits electron transport, cytochrome oxidase. It's not good. It also inhibits LDH, lactate dehydrogenase. It'll cause a loss in the ability of the cell to make energy, and it'll also cause an increase in oxidative stress. And children often will have these problems. Children with autism will often have peripheral markers as disturbances in mitochondrial energy metabolism. For example, they'll often have elevated lactate in their blood. Okay, a few other things about F minus. It's associated with inhibition of the thyroid. It's thought perhaps because they're both in the same vertical column of the periodic table, the halogens. Okay, F minus, chlorine, iodine, and bromine. Okay, also, don't let anybody tell you otherwise. F minus has been long ago proven to lower IQs. This is one study from China where they studied a whole bunch of patient groups, and what they found, 16 patient groups, a lot of patients, they found that children who live in a high fluoride area, so they're drinking that stuff, they have 5 times higher rate of developing low IQs. Like I think I heard somewhere else, 52 out of 55 studies showed lowering of IQ. I wonder who funded the other 3. Okay, Al is toxic to mitochondria, cytochrome C. Also nitric oxide is too, and that's the nitric oxide from inducible nitric oxide synthase. So those are other things that inhibit cytochrome C oxidase. It's competing with oxygen, okay? That's kind of interesting. Al and F minus forming complexes like AF4, they mimic the structure and size of a phosphate, and because of that, they'll bind to ADP, adenosine diphosphate, they'll bind to GDP, and then they can cause dysfunction in those enzyme systems, you know, the ATP and the GDP enzyme systems, and that can be a real problem for the cell. The Al binds more strongly than does magnesium. You always need magnesium for ATP reactions, but unfortunately Al, when it's present, has a way of getting itself in there unwantedly. Al3 neurotoxicity is greatly enhanced by low-dose fluoride. So what I'm trying to say is this is tap water, okay? Tap water. The tap water is toxic, okay? And a lot of people use tap water to reconstitute their infant formula, okay, for the baby. So the baby's getting exposed to this at a young age. That is not good for the baby brain. Okay, I kind of talked about this, you know, how Al3 will mimic phosphates and it'll disrupt enzymes and ATPs and GDPs. It also, like I said, binds cytochrome C oxidase, the inner mitochondrial membranes, succinate dehydrogenase and Krebs cycle. So it messes up energy metabolism. It drops your energy level. And that's why I also sometimes talk about food and mood. Something that's dropping the energy level of your brain, that's going to decrease your mood. You're going to see, gee, I feel tired, something's wrong, I just don't have my usual zip and energy. Here's another paper about neurodegeneration mediated by nitric oxide from activated glia, inhibiting neuronal respiration. Okay, so what happens is you'll have an activated microglia. It'll turn on its enzyme inducible nitric oxide synthase. So inducible nitric oxide synthase is bad because it stays on for way too long, because it needs to have this long-term job of killing all these bacteria if it thinks things are bacteria. Versus endothelial nitric oxide synthase is only briefly on and off. It does what it's supposed to, whereas this one stays on, can be, you know, sometimes you need it to clear out an infection, but in a healthy phase, when it's inadvertently turned on, it can stay on a prolonged amount of time. That excessive nitric oxide, the gas, diffuses over to the neuron, and it'll inhibit the mitochondria, cytochrome C level, and that'll then decrease the ability of the mitochondria to make ATP. When ATP goes down, you can't pump calcium out, and you're prone to excitotoxicity, which could cause the cell to die. Okay. Well, here's a nice illustration of. So here's the activated glia, microglia. It becomes activated and inflamed by something: stress, diabetes, sleep deprivation, head trauma, inflammatory cytokines like IL-1 and TNF-alpha, and also could be bacterial endotoxins, LPS, LTA. For whatever the reason something activated, so it'll then turn on inducible nitric oxide synthase. This makes too much nitric oxide. It's a gas, so it diffuses over to the neuron, and it can then do a lot of damage. The big problem is it inhibits the mitochondria at the level, the inner mitochondrial membrane, cytochrome C, and it's competing with oxygen. And then the neuron is damaged by that, the mitochondria is damaged by it, so the neuron can't make as much ATP. If you can't make enough ATP, you can't keep phosphatidylserine from becoming externalized, and you can't also pump your calcium out, so the neuron's screwed. It's going into apoptosis. Something called diminished brain resilience syndrome by this lady here, Stephanie. She's also wrote a lot about GP, and she talked about the poor nutrition of kids, and then they get repeated head traumas. It causes a lot of brain damage. She's a big believer that people shouldn't be wearing so much sunscreen for all the toxic chemicals in sunscreen. I agree with that. Okay, these are some things that can activate the microglia. Al, we talked about that. Omega-6 toxic aldehydes, you know, hydroxynonenal, glyphosate, beta amyloid protein, LPS and LTA. Al increases your risk of fatty liver. It also increases your leaky gut. There's some things like citrate, GP, and F- that increase Al's absorption in the gut. Okay, and again, she called this diminished brain resilience syndrome. So we talked about our microglia. There's resting, ramified, with the branches coming out of them, like an octopus. There's alerted microglia or primed, and then there's chronically activated microglia. They can be temporarily activated and turn off, or they can be chronically activated. The more activated they are, the higher they keep releasing all these inflammatory chemicals. Again, remember, they're trying to sterilize the area around them. They think it's treating an infection, and that can cause a lot of damage to the adjacent cells. Diminished brain resilience syndrome is that lady scientist, she's basically saying that, you know, kids will get one contusion, and then because their nutrition is so poor and they go back to playing their sport so soon, get another contusion, and that can cause a very prolonged activation of microglia and cause a lot of brain damage. So it's like if you have a concussion, my advice would be you want to sit out from playing any type of head trauma sport much longer than most people would think, and really ask yourself, why are you playing a head trauma sport? Because I know a lot of head trauma sports are fun, like football and stuff, but it's still pretty dangerous for your brain. Okay, magnesium lowers the seizure threshold if you're deficient in magnesium, so you don't want that. It'll also, you know, predispose you to being anxious, and it'll increase your risk of excitotoxicity because normally that magnesium sits in the NMDA receptor, preventing it from opening up. So it's pretty important. Okay, so anyways, that's all I had. I thought that was pretty interesting because this concept of immunoexcitotoxicity, the way that inflammation and multiple things activating the microglia can get sort of, again, a vicious cycle going where the microglia starts thinking there's an infection it has to treat and it starts destroying the surrounding neurons in its vicinity, and that repeated chronically in multiple areas causes a loss of neurons and brain damage. I mean, the brain's got a lot of neurons. It's got about 100 billion neurons, so it can lose a few here and there, but you don't want to be losing them unnecessarily, and eventually you'll start losing ones that have, you know, good memories on them. So anyways, I hope that was helpful.